Welcome, everybody. I think we should get started so that we can spend as much time with our wonderful speakers as we have. Welcome to the session on understanding, what is our title, Phytobiomes to improve agricultural productivity. I should have read the title first. If you look at the header in the program book, I just want you to acknowledge that it's actually incorrect, and the committee that's sponsoring this is the APS Public Policy Board, and I'm sure most of you have heard at this point how the Public Policy Board has set forth a goal of advancing our understanding of phytobiomes, that being all of the microbes associated with plants on the surface, the inside, and around the plants. So in this session, we're hoping to highlight some of the current exciting research in this area. And in particular, we've invited speakers to come who will address questions about how phytobiomes can provide insights into plant disease and plant disease protection, how the microbes in communities on plants interact and communicate with each other and communicate with the plant. The field of microbiomes has been greatly advanced by a lot of funding that's gone into the microbiomes that are associated with animals. So we've invited a speaker to come and talk about that research, and from that we're hoping to understand what lessons we can take from the research that's going to promote our understanding of phytobiomes research. And lastly, what are the opportunities for industry and partnerships with industry to translate discoveries in the phytobiomes area into improvements in crop productivity, because it's that translation that ultimately will benefit agriculture. So with that, at the very beginning of the session here, I would like to really thank our speakers for their willingness to spend the time to get here. Most of them have come from great distances to be here with us today to share their work and their ideas. So with that, I'd like to introduce the first speaker, which is Dr. Julia Vorholt, who is at the Institute of Microbiology at the Swiss Federal Institute of Technology in Zurich, Switzerland. So, Julia.
Thank you very much, Gwyn, for the invitation and the opportunity to participate in this symposium and be the first speaker here. It's really a great honor and pleasure for me to be here and share with you some of the insights that we have generated in the last years. So I think really one of the very exciting insights that came out through the work of various labs, including ours, is that the phytosphere composition, the microorganisms that colonize the leaf surfaces, and this also holds for the rhizosphere, are not just random communities of organisms, but they are, in fact, if you, for instance, sample in different corners of one field, you find very similar strains, but also the community composition is very similar. So that means there must be underlying structural principles that we can now address and try to understand what are the basic mechanisms that are responsible for the community composition that we see. So in fact, the phytosphere, as you know, is a huge environment. So it's estimated to be 1 billion square kilometers. So this roughly equals twice the Earth's surface. So this is enormous. And more surprising that we don't know that much about the microorganisms that are usually colonizing healthy plants. So this field has been very much dominated by looking at pathogens, but this is now changing, so there's more and more interest to understand also the role and importance of commensal microorganisms and to understand also their importance for plant health and also plant growth. So what can we anticipate for the organisms that are able to cope and to deal with this environment? So on one hand, we can expect the organisms must be resistant to and have to cope with UV radiation, with temperature shifts, with desiccation, and also, as you can imagine, all these conditions might change very rapidly during one day, and also in terms of the perspective of a microorganism, also unpredictably. In addition, also this habitat is oligotrophic, so it's low in nutrient supply, so the organisms we can expect are competing for the nutrients that become available, and if they are sitting on the outside, here's the cuticle, so there are some nutrients that are leaching out through the surface, and we can expect the organisms are competing and are encountering microbial interactions to withstand in this environment. And as you see here, the bacteria are colonizing predominantly the grooves that are formed at the junctions of the epidermal cells. So what are the basic questions, the approaches? So on one hand, we are doing environmental studies, so this is here on the left side, where we go out and sample and generate data from which we then also generate hypotheses that we can then test more experimentally. And this is shown on the right side. So we are also using synthetic community approaches to mix different isolates and then to ask specific questions on mechanisms of importance of proteins or also on microbial interactions or plant microbe interactions. And the fundamental questions I'm going into now is the question, which type of organisms do we find in the phytosphere? How do they adapt? And you will see we are linking these two questions in the approaches I will introduce in a minute. I will also discuss a little bit the question, what is the role of the normal plant microbiome also for the benefit of the plant? And can we identify plant genetics factors that are involved in shaping the bacterial community composition? So I will start here on the left side, and the approach that we have decided some years back, really, when there was no kind of this approach is done yet, we decided it would be interesting to take the entire community of microorganisms, and you will see we focus on bacteria, to learn what are the abundant proteins in these communities as a proxy to what matters to organisms. For the organisms. And the major advantages of this approach is we get information on gene expression, we can keep environmental conditions, microbial interactions, until the moment that we really capture the microbial communities, and this approach is cultivation independent, so we can also learn about organisms that we do not have yet in culture. And we have started this approach really some years back when we also didn't know exactly whether this approach with rather complex communities would work. But it did, so that we can now also compare what we learn from the phytosphere microbiota from different plants. And you see here we have taken material from Arabidopsis plants, we have taken microbial communities from soybean, white clover and rice, for which we also have taken not only phytosphere samples but also rhizosphere samples also to compare those. What did we learn? As I mentioned, we combine the questions with this approach of metaproteogenomics on one hand to ask who's there, but also what are they doing, and to start from the community composition first here, a refraction analysis, where you see in principle here on the x-axis the number of sequence clones against the operational taxonomic units, and you see that the diversity that we find in the phytosphere is less diverse than, for instance, in a soil, which is also what we expect, but it's reasonably complex, is comparable, in fact, with also the diversity that we have in the human gut system. So another question to ask, who's there? We can do this, of course, by 16S RNA amplification, but also by using and inferring phylogenetic information from metagenomics information. And you see here this tree of life, which is pre-constructed on completely genome-sequenced organisms. And you see some organisms are dominating, so Bacteroidetes, Actinobacteria, but above all, you see here all these are protobacteria on the left side. And if we then zoom into, we see there are a lot of alpha protobacteria. And among those are, for instance, methylobacteria. So these are bacteria that are famous for their ability to use methanol as sole source of carbon and energy. And in fact, it's very easy to demonstrate the presence of this bacteria by just taking a minimal medium which only contains methanol as the carbon source, and then you press a leaf onto this minimal medium, remove the leaf, incubate the plate, and then you see then after some days all these pink colonies here, which are all methylobacteria. So you see here, these are clover leaves. This is soybean, and I could go on forever. I'm showing you the slides, so they're really widespread and they're everywhere. It's not what you think it is. Besides Methylobacterium, also we noted there are other bacteria that are quite dominating, which are shown here, and these are Sphingomonas bacteria, at least on all the dicot plants that we analyzed. So they're also highly abundant, which was not so clear from studies before. And I will come back to these two different groups of bacteria because those are the ones that we found most consistently, and we have gone into more detail to understand also the adaptation. So now moving on to the proteomics level, as mentioned, we did metaproteogenomics to identify the proteins to learn about the functions which matter to these organisms. And what you see in this triangles are all these dots which represent protein families. And the interesting information is in the position of the dot. And you see there's a lot of consistency. A lot of these dots cluster towards the middle of this triangle, suggesting that not only the organisms are similar, but also the abundant proteome is very similar in these communities from bacteria on different phylospheres in principle. This is different if you compare, for instance, the phylosphere with the rhizosphere. You see here more specific adaptation to the organs and to the specific adaptation required for root adaptation versus leaf adaptation. Another way to look at proteomes is also to look at them sorted by genus assignment. And as I mentioned, Methylobacterium sphingomonas are very important or abundant based on genomics information, but also we identified most of the proteins belonging to these two different genera. This is just the top 20 list. What did we learn? For Methylobacterium, for instance, we saw a lot of enzymes which are required for the ability of the organisms to use methanols, sources of carbon energy, so methanol oxidation in principle. And when we mutated this trait in Methylobacterium, we saw also a lot of loss of fitness. So that means under competitive conditions, methylotrophy is important for this organism. For Methylobacterium, there's just one side remark here. We could only identify all these proteins because we had metagenome information. Otherwise, we would have not been able to identify these proteins with the shotgun proteomics approach. So in this case, really, the metagenome was highly important, which meant there were no closely related strains also in the public databases of organisms completely genome sequenced. So here, really, the metagenome helped. And what we saw were, for instance, a lot of so-called TAN-B-dependent receptors. So these are transporters in the outer membrane, which scavenge, for instance, are involved in taking up different carbohydrates. And we can show this also. So if we, for instance, inoculate plants with different bacteria one by one, and we look how many sugars do we detect, then we see in the presence of Sphingomonas a depletion in the sugar concentration on the leaf surfaces, while Methylobacteria are not touching the sugars in principle. Another point I want to make here is that also we looked in the metagenomics information more closely to see whether there are other maybe genes or potential for genes for new traits. And in fact, photosynthesis proteins are difficult to detect in proteomics, so this is also why we had to look here. And we noticed a lot of genes which are showing a potential for photosystems also in phylosphere bacteria. And we were wondering, can we identify which are the organisms and can we monitor whether organisms would produce a photosystem during colonization? And in order to do this, we can use infrared fluorescence. So all the bacteria that you see now shining up in red produce bacterial chlorophyll during plant colonization. And we were wondering, can we specifically learn which are the organisms that are able to produce the photosystem during colonization? And in order to do this, we used a novel technology that was recently invented at ETH Zurich, and we are teaming up with the colleagues towards developing this tool towards biological applications. I'm not going into the details here, but this is in essence an atomic force microscope. It's manipulated the way that it becomes a nanosyringe, which is force controlled. So we can watch objects from below by a standard inverted optical microscope, and we can manipulate our samples from the top. The simplest application you can think of for this type of technology is a pick-and-place technology, in which we approach individual cells, suck them to the opening here, and then move them and also release them again. That means within one step we can selectively pick individual cells and then try to, for instance, proliferate them or do other technologies. And when we did this with random bacteria from leaf washes, we noticed that we can, in fact, by taking one medium, because one cell you can only put on one medium, obviously, but we could cultivate 2/3 of the bacteria, which was against the literature saying that maybe we are able to cultivate 10% of the phylosphere population. So this number is much higher, which means also we can now more systematically also learn which type of bacteria are colonizing the phylosphere. And so back to this picture I showed before, as I mentioned, we were interested to learn which are the bacteria that are producing the photosystem in situ. So we picked those bacteria and we isolated exclusively, again, methylobacteria. And what you see here are different isolates. And you see also the photosystem is expressed to different degree here. It depends, for instance, on the growth medium that we use to cultivate the organism. So there is also a lot of intragenous diversification within the methylobacteria. So up to this point, I mentioned methylobacteria are organisms able to use methanol. They also benefit, or some of them at least, might benefit from sunlight. So this is one strategy, a very specialized strategy if you want, whereas Sphingomonas seems more to be a generalist organism that is in principle maybe patiently waiting that nutrients become available at the outside of the leaves and are then consuming in particular different carbohydrates. So we can really see that different bacteria, genera in principle, have different traits how to adapt to this environment. And we can now ask, are there also shared traits maybe that, among these two genera? And in fact they are. So in principle, what we see here is also, what we detected is a lot of proteins that relate to stress responses. So a lot of proteins that help the bacteria to protect against challenging conditions. And the serendipity finding that we made some years ago when we did the very first proteomics approach was to discover a regulator which has a sigma factor-like domain, which is fused to a receiver domain. So that is unusual in the bacterial world. So we mutated this regulator and saw that bacteria are not able to colonize the plants anymore. So we hit apparently something very important, and we then noticed that this regulator is responsible for the induction of all the stress proteins. And we see this also on the phenotypic level if we dissect stress responses. As one example here, desiccation, you see a Phi-R mutant here in this dilution series is not really able to cope with this condition anymore. And this holds for methylobacteria, but also for Sphingomonas bacteria. We have also severe phenotypes for other types of stresses, such as UV radiation, salt stress, temperature, oxidative stress. So this relates back to the beginning, what I introduced in the beginning, that this environment for the bacteria is highly challenging, and they must be able to adapt to these changes and to all these harsh conditions in principle. But it's also very much reminiscent of the concept of the general stress response, which has been worked out very extensively, for instance, in E. coli and related organisms. Sigma S is the master regulator of general stress response, sigma B in bacillus and other gram-positives. However, there are no orthologs of these regulators in the alpha-proteobacteria, but interestingly, Phi-R, so the regulator that we discovered, is exclusively present in all the alpha-proteobacteria, and therefore we postulate, and this is meanwhile also confirmed, this is really the general stress response in the alpha-proteobacteria, which also comprises organisms such as all the rhizobia or agrobacterium and other important organisms, many of which live in fact in association with hosts. So it's not only the adaptation to the phylosphere, obviously, but adaptation probably to the selective environment these different organisms are living in. So the other group of organisms I also wanted to introduce are Pseudomonas bacteria. So they are also present in the communities, but more sporadically, I would say. So you see here in one of the samples, so MB1, there was quite substantial presence, or we detected the proteins, where in the other samples not too much. So this really goes in line also with the idea that a pathogen might really be there sporadically coming through wind or other infection ways and then eventually proliferate. And what we saw among the top 20 list as an example is, for instance, a flagellin, which is consistent with the literature for which we know that these bacteria are actively, they're motile and searching for nutrients and also trying to enter into the apoplast of the plant. And this brings me now also to the point and the question I raised in the beginning, and how far is also the plant microbiota important to help the plant against protection against pathogens like potentially Pseudomonas bacteria? We can ask this and address this question, and in order to do this, we thought we should again do an experimental approach in which we use the gnotobiotic system and then ask in how far does it matter whether different commensal bacteria are present for the infection process. With the idea behind that maybe the plant has an interest to have a certain microbial community that helps to protect it as an outermost shield of protection against these pathogens. So what do? We mix different, as you can imagine, we use those bacteria that were most consistently found in the phytosphere. So here this is a mixture of different methylobacterium strains, and you see it doesn't matter whether a methylobacterium is present or these are xenic plants. After 3 weeks, the plants are essentially dead. However, this is different if we pre-inoculate the plants with Pseudomonas bacteria. So you see here after 3 weeks of infection that the plants still look healthy and also the pathogen numbers are greatly reduced in contrast to xenic plants. So apparently Pseudomonas bacteria have an effect against the pathogen, against the Pseudomonas that we took as a model here. So the next question was, are all the Pseudomonas bacteria protecting against, helping the plant to protect against Pseudomonas, or are there just individual strains? And what we noticed that all the isolates that we had from the phytosphere were protective, whereas Pseudomonas strains from other habitats did not show a protective effect. But interestingly, it's not a question of colonization ability per se in this model system, because also this strain, as an example, Vitici here, is colonizing the plant equally to equally high densities. So that suggests that there's indeed a genetic basis for this protection mechanisms. And this is what we are trying to find out currently, what is the mechanism of protection and the mode of action of the Pseudomonas bacteria. I don't have the answer yet, but I'm showing you a little bit how we are trying to go for this question. And in fact, we have decided to do an unbiased, in a way, approach in which we look for mutants that have lost the ability to protect against Pseudomonas in this model system. And this is a proof of concept where you see here some plants in this 24-well plate system here that have not been inoculated with one of the protective strains, this FR1 strain. And you see then in this case the pathogen is multiplying quicker in principle, and we monitor pathogen presence and abundance by luminescence because we use a lux-tagged version of this pathogen. And of course after some weeks then we can also monitor plant disease symptoms. And thereby we screened now 5,000 mutants, and of course we have to test them one by one. So that's really a lot of work, but we have identified some mutants that really look interesting, that have lost the ability to protect. And what comes out so far from the analysis that we have, that many of these mutants in principle show intermediate phenotypes, so that might indicate that there's more than one mode of action that helps to protect the plants via Pseudomonas bacteria. But as I said, we are still currently working on this. We are doing double and triple mutant to really try to understand the mechanisms here. So in the last couple of minutes, I would also briefly go into the question, are there plant factors that affect the microbial community composition? And again, we know the environment has an effect on the community composition, and we wanted to reduce variation in the system, and this is why, again, we used the gnotobiotic approach and inoculated the plants with synthetic bacterial communities. So we used different Arabidopsis mutants, which have defects in different traits, and we also use Arabidopsis thaliana accessions, and to ask, are there shifts in the community composition, which we monitor by RISA, or are there shifts in the bacterial abundance in principle? And what we saw as one example here, that are some mutants, in fact, that show shift in the community composition, like this LUX mutants, but also PEG mutants, so there are differences. And these are all the mutants, the plant mutants that we tested. And the ones here on the left side are the ones that we could really reproduce, that they show shifts in the microbial community composition. And among the candidates here that we have are those affecting the cuticle of the plants, also defense signaling IN2 showed shifts. But interestingly, also a number of accessions showed great differences with respect to the community composition. And this is illustrated here also in this slide, where you see that in principle different subpopulations of the community were going up or down, and also the total community was changing. So I think there's really this is a great resource to use accessions in the future also to try and understand and identify new plant genes that are involved in shaping the communities, which might eventually really also be important for the plant if there's an interest to have a stable commensal population, which eventually also helps to protect the plants. And with this, I would like to conclude. So I mentioned phytosphere communities, that a few phylogenetic groups predominate in the phytosphere of the plants that we analyzed. We have seen there are different adaptation mechanisms. Overall, there is consistency in the metaproteomes, but we also see that different genera apparently exhibit different strategies to adapt to this environment, but they also share traits, as I introduced, like the general stress response. We have shown in a model system that Sphingomonas bacteria are able to protect against Pseudomonas, but we have also shown this works against Xanthomonas bacteria. And for plant factors, we were able to identify some mutants of Arabidopsis that showed shift in the communities, and I think it will be a great resource to use also accessions to identify novel genes involved in plant microbiota interactions. And then I also, during the talk, mentioned that we have principles seen that we can cultivate quite a lot of the population of the phytosphere, and this is why we are currently working on a comprehensive strain collection that can be used also for synthetic communities approach, and that we'll be happy to share. We are currently really going into the process of sequencing 3,000 or 4,000 strains based on 16S and also representative exemplars with whole genome sequencing. And with this, I would like to thank the people involved in this work. Christine Vogel is a PhD student who's interested to uncover the mode of protection by Sphingomonas bacteria. Daniel Müller is currently working on the Arabidopsis phylosphere collection. Florian Riffel is working on metabolomics in the phylosphere, and Natascha Bodenhausen worked on the screen towards identifying plant genetic factors that shifted the community. Then in the end, I also want to advertise a meeting that Gwen Betty and also Steve Lindow, Jan Levaux, Walt Mehaffey and others are organizing next year in Switzerland. So they are very welcome to check this website to have more information. And of course, we would highly welcome people joining us for this meeting in Ascona next year. And with this, I would like to conclude and thank you for your attention. And I'm happy to answer questions. Thank you very much. Yeah, so the question was whether I think that if you use different soybean variants, also the communities should shift. This is what I would expect. We have not done this work with soybean plants. We are focusing really on Arabidopsis because there also the tools are developed then to identify the gene, obviously. But I think it would be interesting to do this with soybean. But I would expect there are differences, yeah. Yeah?
The technique that allows you to pick up individual cells for cultivation is remarkable. The question is, what fraction of the individual cells that we're able to culture had not been cultured before by leaf pressing or other methods?
Yeah, I mean, this is what we are currently systematically doing to isolate all the bacteria. But again, because this technique is nice if you have just a few to pick, but if it's really a lot, so we are using now classical approaches, but paying really attention that we have them isolated on different plates, so not having them close together, because I think what happened a lot if you isolate just by dilution is that they inhibit each other, and this is a problem. And from what we have now done with classical methods is that I think we are at this number that we can capture 2/3 of all the diversity.
Well, you said 65% of the cells you picked up cultured.
Yeah.
Were any of those new to science or new because there's been a lot of things cultured off of it?
I mean, in the way new to science, because nobody has really done this so systematically. So if we look at all the strains that we have, we see it reflects really very well the cultivation-independent approaches. So I think there are also many organisms that we don't have in culture that are completely new. But people haven't, I think, really tried hard to get some of the subpopulation, you know what I mean? It's just more a consequence of how people so far have approached non-pathogenic organisms, I would say. But it reflects really perfectly the cultivation-independent approaches. Mm-hmm.
Technical question about the effect of the Carbadox mutations on the phytosphere microbiota. How did you create those communities? Did you grow the plants outdoors? If so, agricultural environment, forest?
Yeah, I mean, our idea was really to eliminate all environmental factors, so we're using a gnotobiotic system, so in magenta boxes. So this is all highly defined, and we have in principle only those bacteria that we use for inoculation. And the synthetic community in this case was a small community of 7 members only, which roughly represents some alpha proteobacteria, Bacteroidetes, etc., which I introduced in the beginning. It will be interesting to enlarge this type of analysis, I think, in the future to make it more complex. But this is how we started this approach in principle. So we eliminate all these factors. So this is really highly controlled. Yeah.
So you showed that the metabolism is fairly similar among a lot of different agricultural species. Have you looked at, and I would expect that those communities and how they operate may be more determined not by plant taxonomy, but by leaf lifespan and other characteristics that would sort of define their habitat. But if you compared these proteomics with evergreen leaves or longer leaves with longer lifespan?
We haven't done that. We have sampled throughout the season at 2 different time points, but there it was very similar. But I mean, just one remark to what you said. In principle, the metabolism of different bacteria can be very different. It's just because the community is similar. You see them consistently. But it might be that one genus is always producing a certain protein which is specific for this genus, and another one is producing something else, has a completely different metabolism, but because they are equally present in this community, we see them consistently. Yeah, sure.
Thank you so much, Julia. I'm going to cut off the questions, and please feel free to come up and ask your questions during the break. I'd like to take this chance to thank her. We have a lot of people standing at the back, and we can take this occasion to see if you want to fill in. There's lots of, not lots, but there's some seats up at the front here if you want to come and try and find one. All right, I'd like to introduce our next speaker, and that is Vittorio Venturi, who is coming to us as the group leader from the International Center for Genetic Engineering and Technology in Trieste, Italy, and he'll be speaking to us today on the rice phytobiome and cross-kingdom communication. So thank you.
Thank you very much, Gwen and Jan, for inviting me to be here. I'm actually a member of APS for many years, and it's my first time here, and I'm really enjoying this heterogeneous group of people, and I think it's a great platform to also nurture ideas. So I'm interested, I've always been interested in my research really in the last 12, 13 years on signaling. And I've been working with quorum sensing, and we all probably know what quorum sensing is, so I'm not going to spend too much time on the introduction. So basically bacteria like to live in communities. They don't like to live as unicellular organisms. So they produce signals, usually tend to be diffusible, and this allows them to coordinate their expression in response to their size. So, you know, if there's a lot of us, we can do certain things that are more beneficial with respect to when there was few of us. So I've been studying a lot of this, and I must admit I've been studying like with my colleagues using, you know, monoculture, monostrain scenario. But I think now we need to really move on to, we need to move on and really we have to think of bacteria as living as interspecies communities. So which is a big challenge for us, you know, that we're studying signaling in our simple, you know, conical flask with our nice strain. You know, we have to think that actually these bacteria don't live alone. They live together with many, many thousands of species that we just saw from Julia. And in addition, in the case that, in my case, they don't only live together with other species, but live near the plant. So an additional challenge is what kind of signaling is going on among themselves, the bacteria, and with the plant. So I think these are questions we need to address in the future. So if we look at signaling, just briefly, and here I just mentioned this gram-negative bacteria. What kind of signals do we have? Well, this slide just shows you some of the signals, and in my view, this is just the tip of the iceberg. Okay, so we're always finding more and more. The winners so far in gram-negative bacteria are the acyl homoserine lactones that I'm sure a lot of you have heard about. The reason I think being is that they are very easy to study and very easy to detect. It doesn't mean they are the most common, okay, but so far by far the most studied. And the system is very simple indeed. Proteobacteria produce a lot of them, produce these acyl homoserine lactones. It's a very simple system. There is a synthase gene. Sorry, it's not so easy for me to see the screen. So they have a synthase gene that codes for an enzyme that makes this homoserine lactone. And a high cell density, these are diffusible molecules. A high cell density, there is a regulator which belongs to the Luxar family, which actually directly interacts with this homoserine lactone. It's very, very strong usually, this binding, sometimes reversible. And this binding allows the protein to become active and bind DNA and switch on gene expression. So it's extremely simple. So I've been doing a lot of this studying in rice-associated bacteria. That's my model plant, rice, because I also work in an institute that dedicates research and attention to developing countries, so I don't have to introduce the importance of rice. And I looked at a lot of bacteria which I think are important for rice, both pathogenic and beneficial. And for the last 10 years I asked the question, do they produce homoserine lactose? Is this community behavior important for living with rice? Okay, I don't have time to go into the details, but I've done a lot of this work. What I want to talk about this morning is that doing this work, as it happens in research, we bumped across two very important species in general. Xanthomonas oryzes is the second most important pathogen of rice, and Pseudomonas, rhizophyll Pseudomonas, we were studying them and we were asking the questions, are they making homoserine lactones? Are they using quorum sensing to regulate important traits, both beneficial, pathogenic, simple questions. And we could see that, doing this work, we could see that actually these Xanthomonas oryzes could not produce homoserine lactones. Cannot produce homoserine lactones, now we know for sure. When we initiated this work, there was no genome sequences. And most Pseudomonas, rhizophyll Pseudomonas, do not produce homoserine lactones as well. But what we bumped across is that the Xanthomonas and the Pseudomonas, they have the Luxar family regulator. So they have the typical, which is a protein of the typical structure of binding homoserine lactones and responding to them. So we thought, this is interesting, what's happening here? So we had two working models. So do these bacteria respond to homoserine lactones produced by other bacteria, sort of like endogenous? This is also called eavesdropping, which is, makes sense because, as I said it before, and as we heard, bacteria live together, many different species live together. Or alternatively, is this protein responding to something made by the plant? So, PhD student, 3 or 4 years ago, actually a little bit more, also 6, 7 years ago, we started working on these two proteins, one from Xanthomonas and one from Pseudomonas, and we asked the questions: do they respond to exogenous homoserine lactones? Again, I have little time, so I'm going through quickly, but I'll be happy to talk with any of you if you want to know the details. But so she started, we started working on this aspect. And here I had a great tip from P. Greenberg, and who knows these proteins very well, and he told me, well, a trick is biochemically these proteins are highly unstable. They degrade, or they form, they're insoluble in the absence of the cognate signal. When they bind the signal, when they bind their homoserine lactone, in this case, with classical Luxars, they become highly soluble. So why don't you use this biochemical trait to see if they bind homoserine lactones? And that's what we did. We purified the Xanthomonas protein and the Pseudomonas protein. We gave it to a lot of AHLs. And in all the cases, in all the structurally different AHLs that are available out there, we could not solubilize these two proteins. So this is indirect evidence that these proteins are not binding homoserine lactones. And then we did the crucial experiment that changed the course of this work, is that instead of providing homoserine lactones, we macerated rice and extract to the bacteria, and bingo, there we could see the protein becoming highly soluble And here, this is solubility, highly soluble when we purify it, and also with antibodies we could see that. So we concluded at that point that there was something in rice that could probably get into the bacteria, bind the protein, and solubilize it, which is not this is not an homoserine lactone. So then we, together also with Chinese colleagues, we see that next to this regulator there is a proline aminopeptidase gene, which in the promoter sequence there is as I'll show you in the next slide, there's a typical classical DNA sequence which usually is recognized by these proteins. And in fact, we could show that this is actually a very important target gene of this protein. And the expression of this gene is regulated by rice extract. You can see here, if we make a promoter fusion in a plasmid and we provide it to the bacteria, and the blue is the wild type, we can see that if we grow in rich medium and we add rice extract, the expression increases dramatically, whereas in the mutant, we don't see this expression. So this obviously was a target gene which helped us prove the concept that this protein is binding to a plant compound. Then we could see that we also at that stage we could begin to see that this protein, with all the genome sequences coming out, was actually very common, this gene, and all the genes had a PIP gene next to it, which had this binding region. So then we did quite a lot of work. Again, I have little time. We did two proteins in Xanthomonas and Pseudomonas. We did microarrays. We did in vivo proteomics. All the stuff that you do if you're a molecular biologist, and then. So we gained a lot of data, and I can just conclude that these two proteins respond to plant signals. In the case of Pseudomonas, it regulates biocontrol, and we also found some target genes. I did this in collaboration with Joy Sloper, also and Christoph Kiel. With Xanthomonas, we did a bit more work on it, and again here we could find several targets, and it's involved in virulence, and it's very important for regulating motility. So we think it's very important in the initial stages of infection when this Xanthomonas comes in and moves very fast in the xylem. Okay, so this work, the message I want to give here of this work is that after we published this, other scientists started publishing, like Steve Lindau, among others, have published similar proteins, basically orthologs in other beneficial or pathogenic bacteria. So, and we've recently written, a couple of reviews to really send away the signal that this is a novel, widespread interkingdom signaling circuit, and now, if we take this OER and we throw it in the data bank, I'm not good with bioinformatics, but also people help me. We find these orthologs in a lot of bacteria. You can see here the rhizobiomes and the Pseudomonas and the agrobacteriums and the Xanthomonas. So the message I want to give you, this is a widespread, communication circuit between bacteria and plants. All these bacteria that we find, these are plant associated. So I think, signaling between bacteria and plants has really been done in Rhizobium and Agrobacterium, a little bit ignored in other plant-associated bacteria. And so the message here is that this is one of them. This is a new signaling circuit that is widespread, and I think is one of many. Okay, so it's something that we have to look into the future. These bacteria have lived for millions of years with plants. So in my view, there's a lot of signaling going on, and we've shown here one of them. Okay. So now, obviously, the critical step, we're looking for the signal. And in my view, it could be that, again, we have orthologs. It could be a family of compounds which are highly related, just like the homoserine lactones, they have some variation in structure. Again, here probably they are, a family of chemically related compounds that these proteins respond to. Okay. Okay. So the take-home message I've given it. So for the second part of my talk, this is all unpublished data, I thought just to, we want to also join this new idea of studying plant microbe interactions. So we looked at rice and we thought let's really look at let's look at what kind of bacteria live with rice, and we thought to focus on endophytes. Okay. Firstly, let me just say that Italy is the first rice producer in Europe. This doesn't mean much. It's only 250,000 hectares, but we are the first producers in Europe. So we thought to look at endophytes. We think endophytes. Well, we all know that endophytes is a bacteria or fungus that lives within plants for at least part of its life without causing any harm. Okay, it's either harmless or beneficial. And as far as I know, I mean, again, here, they've been found in basically all plant species. And there is a feeling that these endophytes could be used as future bioinoculants for biostimulants or biocontrol agents. So we thought, again, here, we thought, let's start really with the simple stuff, very basic. So we went to rice fields in Italy, and we thought to start, rice is grown in dry conditions or submerged conditions. So we took the two conditions, the 3 stages of the plants, tillering or growth stage or pre-ripening. And then we took several plants for each of the stages, and then we looked at roots, stem, and leaves. So very simple, and we start isolating culturable bacteria. I was surprised the amount of work this actually is, for us molecular biologists that like to look at a protein and do all the gene expression profile and the mutants. 1,318 isolates, you can imagine isolating each one and growing them and so on. And this is just, again, we start simple work. We find these guys. What are these guys? Again, here we found, we looked at genera. We found several genera. Some, we only find in the dry. Some we only find in the submerged. Most of them are in common. Again, we do a distribution, and we find, that the distribution is similar. There are some guys that are found more in submerged, some dry, some more in stem leaf. So we're beginning to see that there are some little trends going on. And we've been funded by, this work has been funded by a foundation that said, why don't you look for, maybe some biostimulants or bioinoculants for Italian rice. So again, I think it's a lot of work. It is a lot of work. We went through it. And for me, it was very important to make sure that they're endophytes, because, when we isolate endophytes, we sterilize the plant, but, you might be picking up rhizospheric bacteria or from the philosphere or whatever. So we did quite a lot of work, and we came down to 30 isolates that we're very happy with, that we can put them back into the plant. So with these 30 guys, we looked at plant growth promotion in the greenhouses, and we have some isolates, especially 3 that look very promising. But with all the 30 isolates, as I'm involved in signaling, we're doing a lot of synergistic experiments right now. We're putting them together. We see if we can see some phenotypes that are synergistic when 1, 2, or 3 more bacteria are present. So we're also doing these experiments. So we're doing genome sequencing, synergism experiments. We're looking at 3 or 4. We're going to look at formulation, and we're looking at doing some field trials. Okay. And at the same time, we're doing this work. We did the metagenomics, and again here, we looked at our roots, submerged dry, the leaves, and the stems, and we could see trends that the big majority is protobacteria, but we do also see some trends that we have, and so we begin to get a picture of what's going on, right? And we would like to reproduce this in rice growing areas around the world, just like Jan said yesterday. I think it's important to do, rice is so important to do it maybe in India, to do it in China, and to do it all around the world, and see if we can make a rice microbiome, and we can see the most important players in the most important endophytes that play a role. Looking also again, we're analyzing. This is all slides that we made a couple of in the last few weeks. Also here we're beginning to see some nice pictures that in the roots we see a lot of streptomyces, whereas we see a lot of Pantoea in the stems and so on. So we do begin to see some trends in Italy, and again, I would like to look at signaling here. I would like to see synergism, and I also would like to see these kind of communities around the world as well, rice growing around the world. And finally, in the last 2 slides, I just, being a molecular biologist, we took one of these endophytes, a rice endophyte that is well characterized, a Burkholderia cururiensis. It's been isolated in Brazil. It's a disotrophic species. It's been shown to be very beneficial and fix nitrogen for the plant. The genome was also, we sequenced the genome by Illumina and pyrosequencing in order to, and then we started looking at gene expression. So also this is, so what I like to do, what kind of gene expression takes place in planta. So what we did the classical, this is the stuff I like. I'm a little bit older. Classical transposon promoter probe studies. We did a mutant bank, and then we looked at plates with and without, rice extract, and look at which genes are going up and down. And of course, we have to do the omics stuff. We did RNA-seq as well in parallel. And we have, with the first part, we found 60 odd transposon insertions, which were clearly differentially regulated in the presence and absence of rice extract. And again, with the RNA-seq, we have a lot of information. But I don't want to go too much into detail because there's a poster on this, all this expression data on this rice endophyte. again, so this, we're also, we would like to, we begin to see what kind of family of genes are expressed more importantly in the plant. So that's it. So the messages I want to give in my talk, that I think interkingdom signaling has been ignored. Has really been studied in Rhizobium and Agrobacterium. We need to focus a lot. I mean, I like to focus a lot on that. I believe there are very important cues, especially with the core microbiome. So bacteria that core microbiomes of certain plants. I think there will be very important cues there to look at, to find what kind of signaling is going on with these bacteria. In my case, in rice. And also we've collected a large endophytic community of rice in Italy, and we want to do that around the world. We have some endophytes that are clearly endophytes. We're localizing them with GFP. We're doing synergism experiments. I think there's interspecies community signaling going on there, and also with the plant. And also we're looking at more close gene expression with some of these endophytes as well. Okay, so thank you very much for allowing me to talk to you about what some of the work in my lab. And I've been talking, the work on OER has been done by Sara in my lab, and now she's working here in the U.S. Sujata has done the work on the Pseudomonas. She's working in Canada, and now also the Bruna here. She's actually here, and she'll be at the poster. She did the work on the Burkholderia crurensis, and Edith Bertani has done all this 3 years of work of collecting all these, this large amount of work collecting the endophytes and doing the metagenomics. And the funding has been from Brazil and from a foundation in Italy that really wants us to look for bioinoculants. Okay, thank you very much.
So your artifacts that you found from lights, how specific are they, and how do you divide up on crops that light?
No, we don't know. We've only... Okay, so the question is... Very nice question is how specific are our endophytes? Have we tried to put them in other crops? The question is we don't know. We've only put them on rice. Maybe the few that we've shown there, the few that we're working with, maybe we can move them around. But from Jan's talk yesterday, it seems that maybe these endophytes could well colonize lots of different plants. There are examples in the literature of endophytes that can colonize. The same endophyte can colonize many different plants indeed. So it's something we will look into.
Make sure these are really endophytes. What's the process that you use?
Well, we want to put them in and sorry. So the question is how do we make sure they're endophytes? And I think that's a very important question. I'm not an expert in isolating bacteria from plants, but we need to sterilize plants, and then you could be ending up with there's so many different components. So what we do is we put them, we infect plants and we make sure that we can recover them from the stem and from the leaves, and then also we put GFP and do some microscopy. Yeah.
I have two questions. First, are you checking if these endophytes are transmitting to the next generation of plants? And second, are you, besides bioinoculant, are you going to look for some endophytes that can help to fight diseases?
Okay, there were two parts to the question. The first part was, do these endophytes can be carried on to the next generation? We don't know. We haven't done that. We haven't done that work. I think that's an important point. The second question is, yeah, sure. At the moment we looked at only, we've isolated these endophytes I didn't mention from healthy plants. We haven't done any, abiotic stress or biotic stress. It's something we will look into in the future. Whether the plants being colonized by these endophytes, by one or more, whether they are more fit against abiotic and biotic stresses. No questions on the first part of my talk. That's interesting. Okay. Okay.
So I know you don't know the signal that is recognized from the plant. Do you have any evidence that it's the same signal from different plant species to different organisms?
We don't have evidence. We've used different extracts of different plants, and we were able to solubilize the protein or activate the expression of the target gene. For example, with the OER from Xanthomonas oryzae, with wheat, with cucumber. So, what I can say that the molecule is present in more plants, but we know that it's very small and and we can extract it with some organic solvents. What we're trying to do when we are purifying, we're trying to purify large amounts of proteins, protein bound to the signal, and then trying to crystallize it. Yeah.
What's the incidence of colonization in fecal mass? You get 100% of the time?
It's good. The few ones that we work with, it's reproducible and we're very comfortable with. Oh, what is the incidence of the colonization of our endophytes? You know, we started with a lot, so the few ones that we're working with, we're very confident. Okay.
Question concerning the signaling. Can you exclude also that the signal is of bacterial origin? Because if you macerate plant material, you also have bacteria.
It's a good question, Julia. Yeah, again, she says, can we exclude the signal that OER or these bacterial proteins bind to is bacterial, of bacterial origin? That's a good question. That was a big concern of mine. So what we did was I took a lot of representative plant-associated bacteria, like Agrobacterium, Rhizobium, and made extracts and tested a lot. You know, we tested over 30, 40 extracts of the, let's say, representative plant-associated bacteria, and none of these extracts could solubilize the protein. So that's the evidence I have. I also, the quantity of the signal, I think, it's maybe if this, like so many bacteria colonize plants, then maybe if it is bacteriology, maybe it won't be enough. Yeah.
With all of the regulators that exist within the bacterial genome, do you have a gut feeling of, one, how many of those regulators are responding to eavesdrop signals, and if so, or if either way, whether you do or don't, how do you get at an understanding of all of those, even for a single isolate, like one of the bacterial pathogens or one of the beneficial bacteria?
So the question was, how many bacterial proteins do we think are involved in interkingdom signaling or eavesdropping? That's a very good question. It's something that I'm really interested in. So one of my future approaches could be that we have some candidate genes. With respect to the homoserine lactones, we can identify the eavesdroppers. It's quite easy. But I'm more interested also to find new ones. So there are regulators out there that we don't know what they're doing. So I'm very interested in the future to look at these regulators and maybe formulate approaches that find the ones that, just like the OER, that bind to plant signals. I think it's something we need to look at. Yeah. Well, thank you very much.
We need to wait until we can switch it. Again, I'd like to invite the people that are at the back to come. There's no seats left. Never mind. I'm so glad that there's so many people here that we've actually exceeded our capacity. I will get us started here. And I'm conscious of the fact that I want to make sure people are able to move from session to session because we're just a little ahead of time. So that's why I'm wasting time as I stand up here. But I would like to, I think we can get started with our next speaker. I would like to introduce Dr. Eric Triplett, who is a professor and chair of the Department of Microbiology and Cell Biology at the University of Florida in Gainesville. And he'll be talking to us today on separating signal from noise in the design and analysis of host microbe communities.
We're ahead of schedule, so I can stop behaving like a New Jerseyan and talk slowly. I will try. It's a pleasure being here. I want to thank Gwen Beatty for the invitation, the organizers for having me here. It's always great to be in Minneapolis, no matter what the time of year is. It's a great city. So I want to preface this by saying I'm going to give you sort of a stream of consciousness of the data we've collected in my lab the last couple of years. And it's on a variety of projects, but all of it's intended to inform us of what the microbiome initiative might look like and lessons learned from the past. Because I've been a player in two microbial observatory projects, and I'm familiar with but wasn't directly involved in the human microbiome project. So I want to give you some lessons learned there. So let's see, how do I go here? Okay. So first, let's talk about the human microbiome project. And I've learned about that recently because of my work in type 1 diabetes. I'll talk about a little bit today. And that is, my perspective of that project was it was very restrictive on the technologies. That is, in medical research, they like to have standard operating procedures, which in most cases makes sense. But it shackled the PIs to only use certain methodologies. And as the technology grew very quickly, they did not. So if you look, for example, at all the genomes that are available for the Human Microbiome Project, they're all kind of a mess. They're all thousands, hundreds of contigs. None of them are closed. Very few are closed. So I think that's a lesson learned. They purposely sought out to see no association with diseases. They only wanted to look at healthy people, but they didn't look at that many healthy people. And they didn't consider the many confounders of the microbiome. That is, comparing healthy people in San Francisco can be very different from comparing healthy people in Miami. Meanwhile, the Microbial Observatories project was started in 1999, and because it wasn't medical, NSF and USDA were allowed to give investigators tremendous freedom to do whatever technology appealed to them, whatever new things they wanted to try. There were no SOPs. And most projects chose long-term sites, which I think is a take-home lesson here. Wherever possible, use long-term sites with lots of metadata so that you don't have to collect it. You don't have to spend the money to do that. But what was sad about microbial observatories is that there was no concerted effort to share data or share samples, unlike the Human Microbiome Project. So maybe that's something you can consider going forward. So there are lots of ways to analyze these kinds of data, and the valuable thing about sharing data is different groups will get very different results from this massive data. And that doesn't mean the results of one group aren't reproducible versus another. It just means there are lots of ways to look at it and lots of valuable ways to look at it. So I think it's important going forward to have data sharing policies and have some clear goals. And I think the Phytobiome initiative going forward does have clear goals. So I'll discuss recent examples from my lab. I want to talk about signatures of land use in soil and how we discovered that. I want to talk about discovery of novel and or important microbes that you fish out as a result of this work. Talk about surrogates of plant disease and how that may be used to understand an uncultured pathogen. And I want to talk about identification of bacterium associated with the disease that was discovered by a culture-independent approach. And finally, I want to finish with the most recent paper of ours, which I think is a lot of fun, The Likely Importance of the Phythobiome. I'm really proud of that paper because I wrote that one entirely myself. That was fun. I haven't done it in a while. It was great. 3 days. Anyway. So the bottom line here is having an interest in culture-independent approaches to identify specific organisms. Community analyses are great. The network analysis you see lately, it's all the rage, is fun. But I like to find those individual organisms I think are the most important, culture, characterize them, and learn more about them. In soil, the hundreds of genera we found by Illumina high-throughput sequencing in multiple sites and lots of samples, we found just 2 genera that appeared to be key, Bradyrhizobium and the ammonia-oxidizing archaean Nitrososphaera. It's not yet cultured. I'll talk about a surrogate of the pathogen for citrus screening. We call it Liberobacter crescens. And finally, an organism we think is associated with type 1 diabetes, Bacterial historiae. So first, to this microbial observatory to learn the drivers of archaeal diversity in agricultural soils. And this was a result of work we published in 2007, where it appeared as though agricultural soils had a lot more ammonia-oxidizing archaea than non-agricultural soils. And we wanted to know what drove that. And the result of that was this paper here, where I give you the citation. And this was work done, finished up by Caterina Zalnina, a grad student who finished my lab this spring, and Patricia de Quatros from Brazil. And we looked at 3 long-term sites. And I think this is an important take-home lesson. Wherever you can, use long-term sites, whether they be LTRs or neons or what have you. But one of my favorite places in the world is Rothamsted Research. If you've never been there, you've got to go. It's a great place. And we used the Broad-Bach experiment, which is an experiment with a fertilizer experiment that began in 1843 and has had continuous wheat ever since then. We also looked at Kellogg Biological Station, the LTR site in Michigan, because it's got long-term site and nitrogen fertilizer trials. And the Everglades agricultural area, because that's an area of interest to me, because its soil is 85% organic matter. It's just way too interesting. And it turns out to have a very high amount of ammonia oxidizers. And the other reason we chose these 3 sites is that they all have, right next to their agricultural plots, they have sites that haven't seen agriculture, in the case of Rothamsted, for about 700 years, in the case of Kellogg for about close to 60 years. And the Everglades agriculture area, as you may know, was early in the last century. A lot of it was drained for agriculture, so that agriculture has been there since about 1910 or so. Okay, so we had long-term metadata available for each site. We had cropping history, soil nutrients, history of nutrient application. And through high-throughput sequencing and all these plots, we had hundreds of genera to sort through. So what we decided to do is look at those genera that were highly correlated with metadata, especially nitrogen. What could we find of all the metadata we take, which genera are correlated with these? And we're particularly interested in those genera that are correlated at all 3 sites. And we found 2. So this is that's a little confusing, but basically if those plots that had the highest amount of nitrososphere were in red, and they were the in red here, these had high amounts of bradyrhizobium. And that bradyrhizobium declined as you had agricultural use. And the reverse was true with nitrososphere, where the agricultural plots, non-agricultural plots are very low in nitrososphere, and with agriculture, its abundance increased. And these 2 genera were in fairly high abundance, anywhere from 1 to 10% of the total soil population. And it kind of makes sense. Bradyrhizobium being a nitrogen fixer, you could imagine it would not be so prevalent in areas with high nitrogen fertilization, whereas nitrososphere, being an ammonia oxidizer, might prefer an environment with high nitrogen fertilization, so it makes sense. So we think there's some markers. The bradyrhizobium here we've since found are most closely related to Bradyrhizobium oligotrophicum, which is found as a free-living nitrogen fixer in Asia and other places. So we think this is a pretty important organism in nature. And so we did box plots, and just to illustrate this, whether it's at at Broad-Bach in England, or Kellogg in Michigan, or the Everglades in Florida, nitrososphere is always higher in agricultural plots, and significantly so. The reverse is true with bradyrhizobium, whether it be Rothamsted in England or Kellogg in Michigan or the Everglades in Florida. In each case, bradyrhizobium is significantly higher in the non-agricultural plots. So basically, I just want to tell you that you can find diamonds in the rough with all this data. And the other thing that's interesting is that these things are reversible. So we looked at the Kellogg successional plots. And if you look at years away from agriculture, so for example, at the time we collected the data, these sites were 58 years away from agriculture. As the farther away you get from agriculture, nitrososphere declines pretty steadily. And the reversibility of bradyrhizobium is much faster. And by 38 years, it's returned to its pre-agricultural levels. So these things are reversible. I thought that was interesting too. But what's really driving their abundance? Both genera were highly correlated with pH. And if you look at the literature in soil microbial diversity, they say the primary driver is pH. Well, yeah, but it's what pH does that matters. So this is the first instance where my major professor was right when he told me to take PChem. I finally used physical chemistry. So I admitted it to him years later, but thanked him for that. So if in all these plots they measure total ammonium, that is total ammonium plus ammonia. And if you take that number, you'll see there's no correlation with ammonia. But if you take that number and take into account the pKa of ammonia and the pH, you can calculate the free ammonia level. And when you do that, the correlation pops right up. So if you just calculate the free ammonia concentration, you'll see a strong negative correlation with bradyrhizobium, which makes sense. A nitrogen fixer should be negatively correlated with ammonia. And the reverse is true for Nitrosphaera, although at low pH, the ammonia levels are so low, it really, your estimate of free ammonia concentration probably isn't accurate. But nevertheless, it looks like of all the metadata there, it's ammonia that's the driver of the ammonia oxidizers, which again isn't very surprising. But here again, it's the value of getting the metadata and the value of finding diamonds in the rough, and you get some interesting things. And of course, once we find an interesting organism, we'll learn more about it. Since we're paid by Florida, we call this Nitrosphaera evergladensis. My grad student, Katerina, was very interested in making an enrichment culture. So she enriched a culture of this to about 50% of the total population. And at that point, you just sequence a PacBio. We're huge fans of PacBio. I recommend it. It's great to get a genome sequence in 3 days. I just love it, closed and everything. So with kind of this Nitrosphaera evergladensis, we learned a bit about it. Enrichment culture was very similar to the morphology of these things in a hot springs in Austria. They're close relatives. You have to grow this for several days before you see an increase in the population, and she measured this by an increase in the amount of AMOA gene copies, and she saw utilization of ammonia and accumulation of nitrite in these cultures. And I think we could have cultured it to pure culture. Slides aren't advancing anymore. I don't know what happened here. I guess we're done early. Oh, there we go. Okay, let's use that. We got the full genome, and they have a lot of genes for urea metabolism, of course, ammonia, oxidation, chemotaxis, and flagella, and we now understand how it uses ammonia and urea. We think we could get it to pure culture if we beat at it hard enough, but Katerina's taking those cultures with her to her postdoc in Berkeley, and she may continue that. A little bit about surrogates. It'd be nice if the phytobiome could help us understand uncultured pathogens and understand and perhaps find surrogates that can be cultured to help understand those uncultured pathogens. They're useful to help you learn how to culture a pathogen. They're useful for valence mechanisms, and they're useful for means for control. Talked a little about each of those. That doesn't work anymore. So some years ago, in 1995, Mike Davis at the University of Florida, then at the Tropical Research and Education Center in Homestead, now he's at the Citrus Research and Education Center at Lake Alfred. He cultured an organism in 1995 from papaya plants, and he was looking for bungee top disease causal agent, and it turns out that organism doesn't cause that disease, and he just stuck it in his freezer and forgot about it. But at some point, he did do 16S RNA analysis, and he found that it was very, very closely related to Liberbacter asiaticus, the causal agent for citrus greening. And he allowed us to work with it, and we worked with him on this, and we first got the genome, done by an undergrad in my lab, Mike Leonard, who's now a grad student at UCLA. The next thing you do, you want to name it. So we got the official name. It took a long time to get deposited in 2 repositories, which you have to do, but now it's available to the world. We call this close relative of Asiaticus crescens. Crescens is Greek for to grow, because we can culture it. And the next thing you do is, of course, is comparative genomics, and try to, does that give you some hints then on how we might be able to culture the pathogen? And so we did the comparative genomics. That's all published now, and we got a couple of hints. What we do when we get these hints, we give them to Mike Davis, and he tries to improve the culturing of the organism. And I think he's made some progress. We told him to add malic acid. That seems to help. We told him to add some vitamins. That seems to help. But we're not quite into culture yet, but I think we're very close. And these are genes in common between the two pathogens, Liberbacter asiaticus and Liberbacter solanacearum, and our organism crescens. And so they have a lot in common, but there's a lot of differences too. But one of the main things is vitamin synthesis. The pathogens seem to lack that. But here again, we're limited, as you all know, by the quality of genome annotation. And this was a real problem in bacterial genetics, I think, that the annotation is still pretty primitive. So that, for example, in the pathogens we see sometimes they don't have the genes for synthesis of a vitamin, and they don't have the genes for transport either. And they both can't be right. So there's something wrong with their annotation. I hope over time that gets better. But meanwhile, of course, we use this surrogate to look for antibiotics that can inhibit the pathogen. And we found 150 antibiotics so far that can inhibit this surrogate. And all the ones we've chosen look like are flow mobile. Okay. Now, identifying microbes associated with disease. Especially useful for diseases of unknown etiology, of course. But what we've learned is that you have to be cognizant of environmental factors that can affect microbial communities. And beware of functional redundancies when identification of a single causal agent is difficult. So let me give you this example of type 1 diabetes. I know this audience doesn't really care about type 1 diabetes, and that's fine. But we were given a bunch of samples from Finland and to try to find bacterial associations with that disease. And Finland has the highest rate of type 1 diabetes in the world, about 70 cases per 100,000. They gave us 1,000 samples, and we looked at this, and we found one species, Bacteria dorii, that is highly, highly abundant, about 40% of the bacterial population, 7 months before they become autoimmune. It's a very clear association we found. But we did not find this in other studies. I'll get that in a second. But the interesting thing about this cohort that they sent us is all the kids were born in the same hospital. They all lived within 80 kilometers of each other. So many of the confounders of the human microbiome were controlled for. They had the same geography, same diet, same water supply, same health practices. Pollution levels were the same. Culturally, they were very similar. So when you control for all those things in a cohort like that, you have a better chance of finding things like this, an association. Whereas we did a similar study with a German cohort and found nothing. But that's because the samples came from the whole country. We were trying to compare a case from Munich with a control in Hamburg, and it was just noise. So I think we have to be cognizant of the fact of how geography affects any microbial community. We see similar results in two other Finnish cities, but not quite the same. So we don't think Bacteria dorii is itself important in this disease, but we think its functions are. And so we'll have to keep an eye out for that. So I hope that informs folks who get into phytobiome research. And we then, of course, were interested in what this genome looked like, and we sequenced the genome from 2 subjects, now 3 subjects, using the PacBiome platform. Just chose samples that were highly abundant for this organism and just sequenced the heck out of it, and we got the data. And just on a whim, I said to Michael, I said, why don't you look at the methylation patterns? Because the beautiful thing about the PacBiome platform, as you know, it's the only one that doesn't amplify the DNA. And it also has a way then of showing you all the methylation patterns in the DNA. You know where every methyl group is on any base of interest. And so I said, well, let's look at that. And we found of the 2 genomes we started with that both genomes had around 20,000 GATC sites. Now, GATC is the motif methylated by DNA adenine methyltransferase. In one genome, all but 3 of the 20,554 were methylated. In the other genome, none of them were methylated. Now, methylation can have a tremendous impact on gene expression. There have been studies in Salmonella and E. coli where the DNA adenine methyltransferase was knocked out, and they showed a tremendous decline in virulence. So methylation has been shown to have an effect on pathogenicity. So here we have a same dominant strain from 2 different individuals, and if all you did was look at taxonomy or even just the genomes, you would say there's virtually no difference between them. Whereas, in fact, the gene expression levels may be very different, and we'll look at that further. And we had very good reviewers of this paper, and one of them said, why don't you look at the origin of this DNA adenine methyltransferase and show where it is in the genome? And that was great. But first, here's the motifs you get back from PacBio that's showing the 20,524 that GATC motifs we had in one, and all but 3 of those were methylated. The other strain didn't have that at all. But anyway, we looked for the location of the DNA adenine methyltransferase. It was very interesting. It was right here within the middle of an inactive phage. So this strain was methylated just because it had happened to acquire a phage at some point in its evolution that has an active DNA adenine methyltransferase. That apparently is fairly common in bacteria. It can happen this way or not, but we thought it was a lot of fun. So that can certainly affect the future, I think, of how we study the microbiome, the phytobiome, whatever. So we have to think about how epigenetics can shape a community and look at that more carefully. One last thing. We just got a grant funded yesterday from NSF. The award came yesterday, and this proposal was led by Pam Soltis. I don't know if you know Pam, but Pam is one of the premier plant systematists in the world. She works closely with her husband, Doug, who's also outstanding. And they put together a proposal where they wanted to study what's called the ancient floristic disjunction. And I know what that was. But what it is Eastern North America and Eastern China share 65 genera in common. And it's not clear why that is. It's not clear what effect that's had over millions of years of evolution. And this plot just shows the geographic distribution of one of those genera, Liriodendron. And so what we want to do, my lab's small part, I'm just a co-PI, my lab's small part of this is to study diazotrophs across the soil, rhizosphere, apoplast, phytosphere continuum. I want to know how diazotrophs change across that continuum. I'm looking for a grad student to help me. If you know anybody who wants to join me, that'd be great. And we chose long-term study sites, 6 in the United States that were either NEON or LTR sites, and 4 in China that were part of the Chinese Ecosystem Research Network, and we have colleagues there to help us. And these sites in China were chosen to match the altitudinal range in the NEON sites. And here are the plant genera involved. And so what I hope to do, again, is find diamonds in the rough. What are the major diazotrophs we see in these communities? How they differ? There's so many questions you can ask. But we want to culture them, characterize them, and sequence their genomes, and not necessarily in that order. Okay. So lots of questions we can ask here. How the organisms vary in diversity and abundance across plant genera, geography, and anywhere within this continuum. Our expectations are that those diazotrophs within the plant will be more capable of avoiding plant defenses than those outside the plant. We had some data, we published a paper on that about a decade ago. And we think there'll be more nitrogen fixers inside the plant than elsewhere. And these are natural ecosystems, so I expect diazotrophs to play a major role. Some will be novel, abundant, and actively fixing nitrogen, and I think we can't help but learn something interesting. So how best to design a phytobiome project? I think you need clear and focused objectives. You've got to choose sites with lots of metadata. You've got to choose long-term study sites, because the benefit from that is just enormous. I believe it's important to use the data to eventually focus on specific organisms of interest that we can then manipulate in the lab and check phenotypes. And try to find as many connections with the plant host as possible. And so if I go back and look at lessons learned over the last 15 years or so, we've been doing this sort of thing, is look beyond the community level and search for diamonds in the rough. Find associations between microbes, the environment, and hosts. Discover and culture novel organisms. Expand the tree of life. Culture, culture, culture. It's a lot of painful work, but it needs to be done. Associate microbes with disease and disease culture from a culture perspective, particularly those intractable diseases we don't know very much about. And I'll finish with saying, don't forget the methylome. So with that, I'll close, and thanks for coming, and I'll be happy to address any questions you might have. Yes?
You could reflect a little bit, sort of a broad question, but you're identifying microbes that are a large-ish percentage and then inferring importance, right?
Right.
We understand that that may not be the case.
Right.
Do you have any strategies to try and get at actual importance of organisms rather than population?
You have to cut it off somewhere. But you raise a very good point because, how do you know what's really important? We just chose an arbitrary cutoff about 0.1% of the population, looked at all those. But you raise a very good point. So, for example, in soil, if I use that criteria, I would cut off most rhizobia. Because most of them may be about 1,000 or 10,000 per gram of soil, and I never would have picked them up. So I think the thing you can do there, if you sequence deeply enough and you have confidence in your numbers, you can identify correlations with all O2Us, and you can do that work. And I think that's worth doing. And it's so cheap to sequence very deeply now. So that's the only way I can think of to do it. Look for the correlations. Anything else? Yeah, Gwen.
On that same note, when you look for the correlations, at least I found it's a lot easier to look for correlations between a single taxon and a treatment. Do you have approaches where you can look at collective taxons?
Yeah, you can use any taxonomic level you want.
But a set. Repeat the question.
Oh, yeah. Can you look at correlations at higher levels, or even community levels, right?
No, mixtures.
Mixtures of strains. Yeah, sure. If you see, so for example, if you see a number of organisms that appear to be closely correlated to a given trait, you can add their reads. You can add up the reads of any mixture of strains you want and try to then redo the calculation. That's one way of doing it I can think of. So, yes?
I had a question concerning the correlation of agricultural practice and the increase in the ammonium oxidizers.
Mm-hmm.
Do you see at the same time also that the nitrite oxidizers would also increase to
So we're asked whether...
Or does it stop then and then you would predict an overproduction of nitrite?
Asked whether the nitrite oxidizers also went up. We looked at anything in the nitrogen cycle, and nothing like that popped up at all three sites. But the data's out there for me to look at again. We do have several genera that would pop at two sites, but you have to focus the paper somehow. So we just focus on something important at all three. But the data's there. It can be done. Anything else? Okay, thank you very much.
We're going to get started. If everyone could take their seats. Hello. Yeah. All right. I would like to introduce the next speaker in our session. If people want to come in and grab seats before we get started. Welcome back to our session on understanding phytobiomes for increasing agricultural productivity. As I said at the beginning of this session, one of the things that we wanted to highlight in this session is the kinds of lessons that we can take from studies that have been done with animal microbiome systems. As we all know, a lot of more money has gone into those systems, and so they have a bit of a head start. And so we want to learn what we can. And so in keeping with that, we have invited Dr. Thad Stanton, who is the research leader at the National Animal Disease Center in Ames, Iowa, to come and share with us some of his work, and actually someone who is working underneath him, Heather Allen, who was going to come and speak but can't be with us today because of illness. So Thad will be presenting some of her work and integrating it. So with that, I'd like to welcome Dr. Stanton to speak with us.
Thank you, Gwen. Dr. Heather Allen did ask me to convey her regrets that she was unable to be here today. At the same time, I want to thank Gwen Beatty, Jen, and the APS Policy Board for the opportunity to talk about Heather's research and also the research of a colleague of her, Dr. Tori Luft. Well, what I'm going to do is start out by describing how we got to where we are today, talk a little bit about what we've been doing, where we're at now, and also very small at the end, talk about where we think we should go. It's a very nice opportunity, having followed Dr. Eric Triplett, to hear very common ideas going forth. There's about 100 trillion bacteria in the animal gut, or in your gut, as you give or take a few million for each person, as you sit in the audience. And basically, many or almost 90% of these are nutritionally very fastidious to grow. And they're all 95% or more obligate anaerobes. And we estimate, or estimates are, that there's about 500 to 1,000 or greater than 1,000 species that exist in the mammalian intestinal tract. All told, if you add up the genomes and the genes, the microbial genomes that are in our guts and in the pig gut are about 100 times greater, 2 orders of magnitude greater in number than the genome of the host in which they reside. These organisms have co-evolved with us, and they have profound impacts for that reason on human and animal health. We know this from studies where that population has been disturbed, for example, by changes in diets, by antibiotic introductions, or by disease. We also know really good studies that started back at Loban Labs in Notre Dame decades ago, that if the organisms are completely eliminated with notobiotic or germ-free animals, there are major differences in the way that animal behaves or the way that animal functions physiologically than when it has its complement of normal, commensal, mutualistic microbiota. So these mutualists are involved in nutrition and digestion of plant material coming in. They're very important in the development of the immune system of their host. They actually provide direct resistance to disease pathogens passing through the gut. And they are more and more involved in the tissue and organ physiology, not just in the gut, but at remote sites. And I was really glad to hear Eric Triplett talking about diabetes. We also know that there's a gut-lung axis, which, at least in mice, reflects their predisposition for asthma in a mouse model. So we really don't know the breadth and depth of which our gut microbiota impacts our health. One other example that I can give you is that there's growing evidence, if not total proof or convincing proof, that our gut microbiota is associated with even behavior through interactions with the central nervous system. So it's a world that's developing and evolving. Now, this is a mission statement kind of the National Animal Disease Center, and that is that an important component of animal health is that food animals should be free from risk of diseases for themselves, hence the National Animal Disease Center, for other animals on the farm, but also for humans. And this is where the Food Safety and Enteric Pathogen Unit directs our research, the people that are part of my unit. One of the disease risks that we hold really important is imprudent antibiotic use. This is a survey which was taken by NOMS a few years ago, and it was a survey sent out to farms where animals that were between 3 and 10 weeks old, the farms were asked to list which antibiotics were in use at that time. You can see that 43% of them were using chlorotetracycline, Carbadox 31%, and so on and so forth. And there are multiple reasons for these antibiotics to be put into diets and fed to the pigs. Disease prevention, prophylaxis, treatment of active disease, but more often than not, for performance enhancement. In order to make animals, swine in this case, get bigger, heavier, quicker. And even better, to have them get bigger, quicker with less feed. Okay, so it's to reduce the cost of management across the cost that it takes to produce a pound of pork. I will point out that in that survey, just so you know, these are antibiotics that are often used in humans, and the resistance cross-reacts. Tylosin and tilmicin are both macrolide antibiotics, which erythromycin is in the same class, and erythromycin is very often used in human medicine. So cross-resistance is becoming a concern, has been a concern now for several decades, depending on who you talk to. Not only are diverse antibiotics used in animal management, but large amounts are used in the United States. And a recent analysis was that an estimated 1,000 tons of macrolide and tetracycline antibiotics alone are added to swine diets for performance enhancement, disease prevention, and therapy. I think it's a little surprised that with these large quantities, they enter the gut and they are going to impact populations of microbiota, regardless of whether it's intentional or not intentional. And this is what we're after studying. It is our belief and the belief of people who have preceded us that we need rational reduction of antibiotic use in agriculture. And our own particular focus on this is that we need effective antibiotic alternatives for animal health, particularly for performance, as well as for food safety applications. So there very likely is going to be a ban in a few years. We would like to see, and we wish we still had companies around that were focused on animal health with the discovery of antibiotic alternatives. And our focus, research at our lab, is focused both on finding alternatives to antibiotic use, discovery, and also testing existing alternatives to antibiotics that are already on the market. So it's a basic problem, and it's a basic problem. This is the question we're asking. What are the effects of dietary antibiotics on the swine intestinal microbiome? And there are two components to it. Of course, the basic research, because we feel there probably are going to be impacts and collateral effects, but also a more practical approach, and that is if we can define what changes, what insights or support of the research for our discovery of alternatives is supported by biomarkers or analysis of what the antibiotics themselves are doing in performance or in health encouraging applications. So it's basic research as well as practical research. Now, ASP250 is the first antibiotic that we tested once we got a sequencer at the National Animal Disease Center. ASP250 is actually a mix of 3 antibiotics. And there are 3 antibiotics. The mixture is used for prophylactic, therapeutic, and at lower concentrations also for growth promotion applications. And very complicated design. We fed a group of pigs ASP250, and we didn't feed a group of pigs ASP250. And 2 weeks later, we went and sampled feces from the animals, and we looked to see if we could tell differences between the microbiota content. This was the strategy. Bacterial cells were extracted to give DNA, and we sent DNA samples to our collaborators at Michigan State University, Jim Teegee's lab. Jim Teegee's lab, Tim Johnson particularly, was a student who had a microarray for quantitative PCR for over 174 different antibiotic resistance genes. At the same time, we looked for antibiotic resistance by metagenomic analysis by sequencing the same DNA that we had sent to Michigan State. We wanted to know who was there, and so we PCR amplified. Our favorite region of the 16S gene is the V1-V3 region for intestinal bacterial identification, and we did the phylotype analysis. We also did something that was a little bit different because very often we overlook what I consider to be one of the more important formers of ecosystems, and certainly in ocean waters it's become apparent that bacteria phage are important controllers of the evolution, particularly of subspecies. And so Heather Allen, this was her postdoc project before she became a permanent scientist with us, and she looked at the viral DNA or the bacterial DNA extracted from the feces. She didn't do it directly. She first went through an extensive purification stage to eliminate the possibility that there was any extraneous DNA. And that purification step included a lot of DNA treatments. And when she did some controls in there to make sure that what we were eventually going to amplify or subject to metagenome analysis did come from bacteriophage or viral particles. So I was delighted because I'm a lifelong culture guy, Eric Triplett commented on in terms of ecology. But we found, when we looked at 86 samples, and we considered them in total, that we got what other people were talking about. The big three phyla were present. The Firmicutes, which are gram-positive anaerobes, and facultative anaerobes, the Bacteroidetes, which are primarily gram-negative anaerobes, and also Proteobacter, which is a kind of a mixed bag. They're gram-negative bacteria, but you can have obligate anaerobes as well as facultative anaerobes like E. coli among the Proteobacteria. First thing that we found was that ASP250 altered fecal bacterial membership, and the ones to look at here are the non-medicated and the medicated 2 weeks after we started feeding ASP250 diets to the animals. The Bacteroidetes, in blue, decreased. The Firmicutes, which are up here in red, looked about the same. We could detect no significant differences, and there was a clear increase in the Proteobacteria in the medicated group compared to the non-medicated group at day 14. What's going on here? Well, we drilled down from the phylum level down to the genus level. And what we found, consistent with the Bacteroidetes, that the Bacteroides decreased, the genus as a whole decreased. And in the case of the, gram-positives, however, which did not change, the Firmicutes were the same. What we found, there were differences within. In other words, even though we didn't see changes based on the phylum level, we did see at the genus level. There are decreases in Streptococcus, Coprococcus, and an increase in this anaerobe that I'm going to talk about a little bit later, Ruminococcus, which is pretty well studied in rumen microbiology for what it does to plant polymers and the breakdown of plant polymers. It's an important player. We saw increases in Ruminococcus. Escherichia coli and the Proteobacteria went up, and succinivibrio, and I have this in the next slide demonstrated. Here it is, medicated on day 4 versus control. This is succinivibrio. We saw jumps in the relative representation of succinivibrio based on 16S sequences, but we were really surprised to see this sort of a quantum leap in micro terms of the Escherichia shigella group genus. E. coli increased from 1% of the population to 11% of the representation in the fecal DNA samples. This was so impressive to us that we actually repeated the experiment, which you don't see a lot with metagenomic studies. But we did repeat the experiment, and we knew how to grow E. coli. So we went back and we did culture-based analyses. And sure enough, the control pigs, unmedicated versus the medicated pigs, E. coli populations were 2 orders of magnitude higher. They were shed 100 times more in numbers, colony-forming units, than were the ones that had not been medicated, not eaten the ASP250. Well, we first talked about this as an E. coli bloom, and then we found out, and Heather and Tori wrote a review article on it, that E. coli blooms have been known to occur in a lot of other situations, particularly when you have a perturbed gut. It occurs during pregnancy. It can occur with diet changes, starvation. You will see increases in E. coli shed from either the humans or the animals. What about antibiotic resistance? Well, we had 2 ways of looking at that. One was with the metagenome, and even before we fed the animals antibiotics, we detected 149 antibiotic resistance genes, different resistance genes. In the case of qPCR, there were fewer, 57. qPCR was much more sensitive, but it was also too specific for our applications. The metagenome complemented and supplemented the work that we got with the qPCR analyses. ASP250 turned out, we found there were 23 genes that varied differentially in numbers between the treated metagenomes and the control metagenomes. Not surprisingly, a number of these were in response to the antibiotics that we were giving. The beta-lactam, the tetracycline, and the, sorry about this, it's not working, and the sulfa drugs. What was surprising to us was this one right here, which was a streptomycin, which also went up. Well, it turns out the aminoglycoside resistance genes are very often carried as hitchhikers on mobile elements. And it made perfect sense to us once we saw how common it was to go along with those other genes, particularly chlorotetracycline. It made sense. We also, interestingly, there were a few genes, resistance that went up, that were more prevalent in the control metagenomes. So it's not just a direct selection that we feel is taking place. There's probably a competition going on between bacteria, not just the antibiotic resistant selection. So as a result of that, you can see increases in resistance or decreases in resistance to certain populations depending on what's going on in the gut. Hey, it's an ecosystem. It's not just one factor that's involved. The one thing we found interesting, and it becomes a biomarker for us, is that there was one gene functional category that went up, and that was for energy production and conversion, which is category C. We found that more frequently in the medicated metagenome than in the non-medicated metagenomes. What about the phage? Well, Heather Allen took some pictures with Judy Stasko of the morphotypes of bacteriophage that are in fecal samples of swine. Basically, every family, and these bacterial viruses are classified according to their shape and the morphology. The Poloviridae, Cyphloviridae, we even had one that she called the tutu virus because it looked like it had a ballerina costume on it. We've seen this one a number of times, but all morphotypes were represented. We also saw enveloped viruses, and these are very likely animal viruses that were carried along in the purification step. Sherwood Kastians at the University of Utah suggested to us a way to calculate the numbers of phage that are in the preparations, the DNA extracted preparations. He said, why don't you count the phage integrase genes that are present, because they're fairly conserved and they're also essential for every bacteriophage. And so we did. We found that there were significant increases in bacteriophage in fecal samples from animals that had been treated with the antibiotics than animals that were non-medicated. Is this important? Well, if we looked at the metagenome or the viromes of those bacterial viruses, a number of them are carrying antibiotic resistance genes. This makes a lot of sense, as I mentioned earlier, that the role of bacteriophage and bacterial viruses in natural environments is really understudied. We know that it's important from a lot of the work that's now coming out with metagenome sequencing, because we no longer need to culture these molecular parasites. A short treatise on Carbadox. Carbadox is not like the other three antibiotics. The other three antibiotics work either on a metabolic function or work on some intracellular macromolecule in bacteria. Carbadox is a mutagen. It intercalates in the DNA just like ethidium bromide does, and it kills bacteria because it creates more mutations than the bacteria can overcome during its growth in the intestinal tract. This was a longer-term study. We were interested in what happens very early on after animals are fed Carbadox, late Carbadox, and in withdrawal. The withdrawal period, because it's toxicity for pork, is 6 weeks before they go to market. So we wanted to see what happened during this late withdrawal period. I'm not going to go into the details for all the ages or the steps that we took in here, but I would like to mention that where you see these dark colors, these are significantly different from the matching group. What was really impressive to us was a 22% increase in the representation of Prevotella genus in the Carbadox treated compared to the control animals. Rosebaria went up significantly, Lactobacillus went down, a mycoplasma, Asteroplasma came up, and Faecalibacterium also increased its representation in the DNA samples. Now, So there are short-term effects due to Carbadox. We also saw long-term effects, even though, first of all, these all went back to normal after the withdrawal period, at least in terms of the representation. The succinylvibrio, however, went up later on. So we're looking at not just short-term effects of antibiotic treatments on microbiomes, but also long-term effects. And I've taken select genera for the sake of this presentation. You can look at the paper and see what other things happened. What's going on here? Well, Prevotella, like Ruminococcus, is a plant polysaccharide and plant recalcitrant polymer degrading organism. Roseburia and Faecalibacterium are butyrate producing genera. Prevotella makes acetate, releases soluble sugars, which then can cross-feed Roseburia and Faecalibacterium, and acetate in these organisms through butyrate is required for butyrate production. So we look at this as kind of a nice, interesting synergism to be looked at further, perhaps a cross-feeding experiment. This was really, just like the E. coli bloom, we're impressed with this Prevotella bloom. Now, E. coli is not hard to grow, but anaerobes, particularly Prevotella, are very difficult to differentiate by culture-based analysis. So we turned to digital PCR. We analyzed the populations of Prevotella 16S RNA genes that were in this DNA preparation. The bottom line was we found no change. The significance of this is that in absolute abundance, Prevotella did not increase. Other organisms had to decrease to make it look like it was more numerous. So this is our comment right here, that you can see changes in populations, but unless you do a little more below the surface examination, you will discover that maybe the relative abundances aren't nearly as interesting or as important as the real numbers that we see. So I'd like to just sum up what we found to date. That is that ASP250 and Carbadox had measurable impacts on swine fecal microbiota. In the case of ASP250, we saw increases in phage production. We saw increases in 23 antibiotic-resistant genes, COG, gene group C, which was energy and conversion. We also saw increases in E. coli, Ruminococcus, and succinylvibrio. Carbadox, we saw increases in Prevotella and succinylvibrio, although I keep in mind that they weren't real increases in the case of Prevotella, but it was a selective appearance of an increase. Other organisms went down. In fact, to get that percentage, probably double that percent of bacteria had to go down, or we wouldn't have seen a neutral, even number. So all of the organisms that are in yellow basically are important in the breakdown of plant polymers. This is from a Weimer publication. Is from Madison. And so in our view, we are asking now whether performance enhancement are some of these magic bullet taxa. We still have a long way to go with Carbadox studies, by the way. We got metagenome analysis, and we're also doing metatranscriptomic analysis of those populations. However, we've just completed a study of a marketed antibiotic alternative which is sold for swine health, improving swine health, and we're starting to analyze the metagenomes of those samples right now. So I'd like to acknowledge our partners, particularly David Alt, Leanne Hobbs, and bioinformaticist Daryl Bales. Electron microscopy, care of Judy Stasko, who's an artist as well as a scientist. And we had a wonderful relationship with Jim Teege and particularly Tim Johnson in his analysis of antibiotic resistance genes. My own group, these guys in the back, Tori Luft and Heather Allen, are the hands and basically 90% of the time the minds in the research that I've talked about today. The other two over here, Benjamin Bass and Megan Bandrick, are focused on those alternatives to antibiotics and their impact on the microbiota right now. So I was really happy to hear what Eric had to say earlier. We strongly believe that even though these are different eras, culture-based, unigenome-based, or molecule-based efforts at looking at ecosystems. By no means do these deserve to be second class to the metagenomics and bioinformatics-based systems that go on. First of all, the culture base is what led to this. We wouldn't have been able to do this without having culture-based analyses of bacteria. Secondly, I had a postdoc in my lab, Uri Levine, who came from Tom Schmidt's lab. He switched from soil to animals. That was quite a jump. And he isolated over 100 anaerobes from the tract of the pig. And we found butyrate-producing organisms. I think it was 4 or 5 of them were new species. 1 or 2 of them were entirely new genera. And in every case, the current primers that are being used to amplify butyrate genes to detect butyrate-producing bacteria would not work on these organisms. So we need to have this to get to this, and we need this to get to this. These things are all complementary and supplementary. And I just, even though I'm getting ready to retire, I have to tell you, for the young people, this is absolutely the most exciting time to be a microbial ecologist, or to even like microbial ecology or read about it, because of all the tools that you have at your disposal. Thank you very much. Please.
I very much liked your point about the importance of absolute abundance.
Correct.
In the picture that we too often focus on relative abundance. My question is, can we use that information to understand infectious dose of pathogens and or the prevention of infectious dose by the use of probiotics, prebiotics, etc.?
Which approach?
Looking at the absolute abundance. So,
Yeah.
for example,
Yeah, you're right, but with over 1,000 species, I think you need a couple steps before that. Oh, repeat the question was, can we use absolute abundance in efforts to look at organisms that are that will protect pathogens or cause infections? And I think there's room for that, but I think we need a little bit more work. Lexiscupum treated turkeys with antibiotics to reduce the populations, and then found that Campylobacter decreased 10,000-fold with a certain antibiotic. She went back in there and discovered that what was in that mixture, or in that population, was a bug known as Microbiomes hypermegaly. We're trying to isolate that organism right now for obvious reasons. So I think you can set up, as clever microbiologists, I think we can set up some screens or selections that allow us to get a little closer to that question. If you have a better idea, I do want to talk to you, though. Please.
Well, I'm very interested in the observations of the changes in the bacteriophage population,
Yes.
as well as the bacteria, and of course those are dynamic.
Mm-hmm.
So do you have any ideas of if there are specific bacteriophage, what most of those bacteriophage, is that at all correlated with what you've seen in the changes?
You would make Heather really happy, because she has data sets that are starting to trace those. I must tell you, even though we work with feces, we're stopping working on feces. We're going upstream. We're looking at the ileum right now because it's a tighter population. We think it's more important for immunity. We have found a lot of phage production at that site. So we're covering our interests as microbiologists with the animal side people that are interested in immunology. Please.
I'm struck in listening to your talk, which was very nice.
Thank you.
It only deals with bacteria and causes of bacteria. What about all the other critters?
No, you're absolutely right. The question is, why aren't you doing more? and he's right. He is absolutely right. It's a lack of our experience. We do see fungal. We see archaebacterium. We need people who are smarter than we are, because unfortunately we're all microbiologists trained in bacteriology, and that is a bias. At least we got into viruses. So give us a little credit, please. Please.
I was struck by your data with the ASP250. Not the comparison of the non-antibiotic treated to the antibiotic treated at 14 days, but even the differences between a day 0 and day 14 and the non-antibiotic treated, there seems to be a significant change in that microbiome. And I wonder is it possible that there's a development of the ecology there?
Oh, no, you are absolutely right. The question is, do we have other explanations for the data that I'm holding back? And the answer is, in the interest of shortness of time, I've not talked about the maturation of the animal. That is really important. We stuck on post-weaning pigs because that's the most serious point in pig raising, when they're most susceptible to disease and when antibiotics, I think, are most used. Once the animals get past that stage, get into the 60, 70, 80-pound range, you don't have to worry about them anymore. They're able to take care of themselves. But that is the vulnerable period. But yes, we do consider maturation very importantly. Thank you.
I'm going to cut off the questions in the interest of time, but please don't hesitate to find Dr. Stanton afterward and ask him further questions. I would invite all of those who are standing to try and find a seat at this time if they would like to sit down. There are seats all the way across the front here. You can move it. Okay. So next, I would like to introduce Dr. Magali Guilabert, who is the Director of Microbiology in the Biologics Division at Bayer Crop Science. And as I had said at the beginning, one of the things that we wanted to do in the Phytobiomes initiative is really emphasize how there's room for industrial partnerships and for industry in translating the discoveries in phytobiomes into increasing crop productivity. And so with that, we're very happy to hear about what Bayer is doing.
Okay.
It's not coming up. Yes. We're set.
Okay, great, perfect. We are all set. But it's not. Oh yeah, here we are. Okay, good. Okay, well, thank you very much for inviting me today. And so Gwen and Jan asked me to kind of give you an overview of how we work with the phytobiome and what the needs are from the agriculture, from our sector, and how can we use the knowledge and the information we generate from that. So, okay, here we are. So what I decided to do was to give you a background about what we view as biopesticides, as well as some market drivers that are driving the use of biopesticide right now in agriculture. Then I was going to touch base on the plant microbiome and really what we mean by the plant microbiome and what is the knowledge and the different use that we can do of the knowledge that we are generating with the phytobiome. I was then going to give you a little bit of a case study of one of the active ingredients we are working on and what the type of data that we generate and what the needs that we still have. And then I'm going to finish by kind of give you kind of like the future on how we see the different products that modern agriculture would need. So first of all, I thought I was going to kind of explain to you what we view as biologicals. So there is really two different categories. So you have, of course, the microbes, and you also have all the plant extracts. So in general, plant extract, and I'm going to highlight the advantages, but also the challenges. So for plant extracts, the challenge for us is if it's, let's say, if it's plant extract from an exotic crop somewhere in, let's say, Central America, for us the main challenge is about production. So we heard yesterday the mention of snake oil when people are talking about biopesticides. So that's, of course, a main issue for us. And we want to make sure that every single batch, every single manufactured product is exactly the same and is very consistent, and it's to avoid that notion of snake oil, right? So we want to be able to predict how this product is actually going to perform in the field, okay? So in general, for us, plant extracts or any type of botanicals is very challenging unless we can use some commodity crops that we can very much grow consistently. So in general, we do focus on microbes, we do focus on bacteria, and we focus on fungi, and what you're going to see missing from this list are gram-negative. So gram-negative are great biopesticides products. They are great biologicals. We've seen great posters, for example, yesterday. However, they do not form spores, so they only form vegetative cells. So for us, it's very challenging to be able to commercialize and to stabilize products for commercialization. So in general, when we think about a product, we have to think about years. So we have to think about shelf life of a product. We use a specifically in the U.S., a specific regulatory path that requires us to have live cells for many years. And the issue with bacteria that do not form spores is we cannot formulate them to maintain their life, their availability. So of course we are working on it. A lot of companies are working on it, but right now it's a main challenge. So we do focus on spore formers mainly. We focus on Bacillus a lot. The reason for that is they are extremely stable. We heard a lot about stability on the phytosphere, on the phytosphere for like UV stability, temperature, pH. Those spores are extremely stable under those conditions. We also have a lot of expertise on how to ferment them, on how to formulate them. We have a lot of genetic tools, so we have a lot of toolbox that allows us to, first of all, characterize those strains to the genome level, but also to the natural product chemistry level, and we can also improve those strains very well. In general, Bacillus are very safe. So we have not had, of course, we don't work with Bacillus anthracis. We work with species that are very safe. We do not have any toxicity issue. The challenge, though, is usually the chemistry is always the same. So if you want to actually go ahead and look for some other spectrum or for a specific pest, for example, that is not being controlled with Bacillus, is very challenging, which is why we work on Streptomyces as well. So Streptomyces, they are kind of like a chemical factory, if you want. They are very good at producing a lot of different natural product chemistries. Unfortunately, they're also very good at producing a lot of toxic compounds. So which is, of course, a challenge for registration for environmental purposes, but also for human purposes, of course. We also work on fungi. So fungi have the advantage over bacteria that actually they are true biological, meaning they can have mode of action that is through pathogenicity versus bacteria, which is through the expression of secondary metabolites, for example. We have a challenge here that I wanted to highlight is, of course, obviously in agriculture, one product is not used alone. It's going to be used with other fungicide, for example. Fungi are, of course, sensitive to fungicide. So we do have to find other ways to be able to use this fungi, usually by means of genetic means. Okay, so I wanted to give you one example of a very successful commercial biologicals. Okay, so this is a seed treatment. This is called, the commercial product is called Poncho Votivo. It's a combination between a chemical and a bacteria, which is a Bacillus firmus. And what I'm showing you here is a yield bump between the chemical alone versus the combination between a chemical and a microbiome and a microbe. So this is widely commercialized in the US. So this yield bump here shows that, first of all, we can push the production of our crop in a very sustainable way by using, for example, microbe. And the second point is we can actually reduce the amount of chemical that we put on in the environment by complementing with the bacteria and maybe get to the same yield that you will have with a higher rate of a chemical. I also wanted to show you the consistency of this specific combination here. So now you have to think about, we are in a lot of different genetic backgrounds, right? If you think about corn commercial varieties, you have a lot of different genetic backgrounds. We are, mainly in the US, but in a lot of different geographical locations. So to be able to achieve 75% of consistency, it's very remarkable. However, we still have 25% that is not consistent. Okay, so in some cases, and Jane mentioned that yesterday, it could be environment, it could be soil, it could be pH. We just don't know. Sometimes those microbes do not perform. And if we think about modern challenges, and if we think about being sustainable, yet pushing our crop production, and if you think about how much space, for example, there is left on a seed, we cannot add more onto a seed. So we are going to have to improve our products in order to close that gap of the 25%. So we all know, agriculture is facing challenges. I think there are some short challenges and there is some long-term challenges. So the short challenges are, they are, for example, us, the consumers. We want to have safer food. We want to have safer fiber. We want to have all of that happening, in a sustainable way. So the regulators, in response to that, are really trying to eliminate a lot of those old chemistries, if you want, that is, for example, toxic for the environment. And there is some long-term challenges, and we heard a lot about yesterday with Dr. Van Ness, for example, where we heard about precipitation in some part of the world. We heard about drought in some other part of the world. So we are going to have to be able to feed a growing populations with less land and under more challenging conditions. So we think that biopesticide or biologicals, microbes, if you want, can actually address some of those challenges. We don't think it's going to be the solution, or we don't think it's going to be the magical solution. We really truly believe it has to be integrated solutions. So we have to on having the best seeds with the best traits possible, the best, chemical possible, and we do believe that microbe can actually have a role here. So I think we have heard, during this meeting a lot about how microbes can actually act on different aspects of the triangle, right? So I talked to you about the production, for example, of secondary metabolite to control against pathogen. We of course know that microbe can induce or can modulate plant physiology and induce host resistance, and of course we know that microbe can actually alter as well the microbiome. So we have a very diverse set of mode of action. I'm not trying to say that microbes can do everything, but they definitely have the potential to do a lot. Now it's our job to be able to understand how those different mode of actions actually expressed under all the different environmental conditions that we are faced with. How can we improve that? How can we manage it and control it in a way to be able to commercialize new products? So, of course, when you commercialize microbes, you think a lot about microbial ecology. Okay, so the rest of my talk is going to be mainly focused on seed treatment and root microbiome. So I just wanted to say a little bit of things about phytosphere or foliar application. So as you can see in this really simple drawing, we have 3 different types of applications for our product. We have foliar, drench, or seed treatment. So Steve Lindau, years back, did some phytosphere colonization work with Bacillus thuringiensis, for example, and showed that Bacillus thuringiensis was not a good colonizer of the phytosphere. And we have found the same thing. So we know that the way that those microbes are going to work as a foliar treatment. So if you think about what we sell, so we make a fermentation, we take the jug that has the live cells with whatever is being produced during the fermentation, we ferment that, and this is what we sell. So we know that the cells are not going to colonize the leaves. We know there is no in situ production of the chemistry, if you want. So we know that it's the chemistry that is present in the jug that is going to drive the activity. So we have manufacturing plant. We have a very, very large natural product chemistry department, and we make sure that we fully understand what it is in that broth that drives the activity. Everything is QC'd. Everything is manufactured. Every single jug that is leaving the manufacturing plant is exactly the same for the CFU level, but also for the natural product chemistry level. So I'm not going to say more on that. I'm going to now focus on seed treatment. So if you think about seed treatment, and if you think about a seed and what you put on a seed, the amount of the fermentation broth that you put on a seed is very minimum. It's like microliters per seed. So now the amount then of secondary metabolites or of chemistry present is very minimum, and the product really relies on the in situ. So the bacteria is going to colonize the seed, is going to colonize the roots, and then by doing so is going to produce in situ whatever natural product chemistry that it needs. So the root microbiome, it's a very simplistic representation of a very complex, interaction. So we heard, Jen yesterday talking about genome to genome talk, either between microbes, between microbe and pathogens, between microbe and plants. So we know of course that the microbes can talk to each other, that they can signal to the root. We know that the roots in response can also signal back to the microbe. There has been some studies done by Dr. Bay, for example, that shows that malic acid from the plant is being induced by beneficial microbes, and in turn is going to be recruiting other beneficial microbes. So there is a lot of studies around signaling. We know very well the phenotypes that are derived from all those different interactions. We of course love those phenotypes if we start thinking about crop production. And as I mentioned before, the issue is we don't fully understand all those interactions, so it's very hard to really manage and control all those different phenotypes if you think about, on a global level. So we heard yesterday, of course, about the gut microbiome. We heard about how it's a platform with new tools and so on for the plant microbiome. And we really truly believe that, the science is now there. And I totally agree with you, for example, saying as a microbial ecologist, it's great to be now, right? Because it is just so exciting. So, we have all the genetic tools to track the behavior and the changes in these complex populations. We have all the physiologic tool to actually look at those interactions, and we also have a lot of natural product chemistry tool where now we can actually look into the soil with really small amount. We can actually track those signals. So we really believe that, and I think it's Dr. Vaness who yesterday was saying if you can measure it, you can manage it, and we really truly believe if you can measure it, then you can improve it, okay? And we can really now, historically, biologicals they really have a very bad reputation. People think that they don't work, they are not consistent, so it's very the time now that we can make this technology way more consistent. So now I'm going to switch, and I'm going to give you a little bit of data around some of our commercial products. So because of time, I am not going to be describing every single studies that we are doing, but I just wanted to kind of highlight what we know and most importantly what our needs are. So Serenade Soil is a commercial product. The active ingredient is a bacteria that is called Bacillus aeruginosa. It's QSC 713. We know that Serenade Soil can actually we have a very nice effect in the field. Of course, it's under field conditions, so there is different, diseases. There's a lot of things going on. We know that we can reproduce this effect, for example, in the laboratory. So what you are seeing here are just, pure soil without sterile soil, so without any plant pathogens. And you are seeing, so they are tomato plants that have been treated with increasing amount of Serenade soil. So you do see a very nice effect here. This effect, you can actually quantify the effect. And we also know that QSC 713, the bacteria, can actually colonize the roots. And we know that this colonization is actually very important for the phenotype that we are seeing. So obviously something is going on that is interesting. So we are, of course, trying to understand a bit better what is the mode of action of those phenotypes. So we know that the bacteria QSC 713 is actually going to interact with other microbes in the soil. We know that QSC 713 is producing natural product chemistries. They are called lipopeptides. They are pretty much cyclic peptide. We know that those lipopeptides can actually be produced in situ, either in the field or under laboratory conditions. We know that those lipopeptides can actually control soil pathogens, for example, so there's definitely that genome-to-genome talk. And we also know that those lipopeptides can also induce systemic resistance, so an indirect effect against plant pathogens or microbes in general. So we also know that 713 is going to express some volatile compounds. I saw some posters yesterday around those volatile compounds, specifically 2,3-butylenediol. So 713 is expressing such volatile compounds. So what you are seeing here are Petri plates, obviously, that have a physical barrier, so the bacteria cannot touch, if you want, the plants, and only the volatile compounds can actually go from one side to the plate to the other. So you can see a nice effect here. What we have used here in a collaboration with Paul Paré in Texas Tech are promoter fusion Arabidopsis lines, which have the promoter of an enzyme that is involved in the starch synthesis, and you see that this enzyme is turned on in the presence of the volatile compounds. We also know that in consequence, the chlorophyll content is higher in those plants, and we can also see higher biomass. Still, in the laboratory of Dr. Paul Paré in Texas, the volatile compounds increase photosynthesis and can also improve iron uptake. So it's just to kind of give you a flavor of. So it's obviously, I truly believe that we are just scratching the surface, right? So obviously here what we are looking is only one bacteria into one system. Of course we know that, we have a community out there, so we have to be able to look, at the bacteria in the field, under field conditions. I like the mention of plant hosts, right? I mean, we have to be able to look at soybean and corn and wheat, right? Because those are where those bacteria are going to be sold. So, we are starting doing a lot of work around, using metagenomics, transcriptomics. We are not forgetting the epigenomics. So, we're also looking into that to try to dissect out all these very complex interactions. So I just wanted to give you, one example of what we have done to actually register new products that have improved traits, if you want, for soil application. So I told you earlier that recolonization is very important. We know that bacterial populations, like a lot of other populations, are not static, right? So you have cells that can be in different stages. In this case, we had, of course, cells that were either in their sedentary mode or cells that were in either planktonic mode. So we biased the population towards the sedentary mode. So we found a knockout into a regulatory gene. It's a contingency loci that allows the bacteria to switch back and forth really fast between those different stages. And by doing so, we are increasing biofilm formation in this specific variant, and by doing so, we are increasing plant growth promotion into crops that we are interested in. So I'm just going to conclude by kind of like the future, right? So we all know about, the triangle. We all understand that, plant genetics, of course, is very important. The genetic background is important. We understand that the agronomic practices are also very important, and as well as the chemical input. But we really truly believe that now soil microbiome needs to also be in the picture, specifically, to address those challenges that I mentioned before. So I just, wanted to kind of, and of course it's definitely not an exhaustive list, but kind of some thought about, the different future products that we could have, and specifically around looking at the microbiome. So one could think about, plant genetics, for example. We could modify plants to secrete some compounds that can alter the microbiome in a positive way, of course. We can also increase root secretions for specific attractant, and mentioned before malic acid, so that could be one one way, for example, to recruit more beneficial microbes. We can think about developing chemical products that can actually manipulate the microflora or that can actually stimulate colonization. And of course, we can also look at new microbial products. So I liked a lot the mention yesterday about, I think composition is really important, but I think function is also very important. So for example, to be able to find biochemical markers, we talked about carbon source yesterday, for example, that could be a way to actually directly screen for new microbes, for example. So I think really looking at what's there, the potential of the community, but also looking at what exactly is being expressed depending on the environmental conditions can also be very important. And of course, as I mentioned before, if you can measure it, you can optimize it, and that's our main challenge, right? I mean, as I mentioned, we have this 25% gap, and we need to be able to improve those microbes. So, in order to perform, so being able to genetically optimize our microbe is also very important. There is some open questions. We talked this morning about endophilic microbes. It's definitely something that we are thinking about. So there was a talk yesterday around modifying the symbiote of the insect that is transmitting the citrus greening, for example. So that's definitely one option. So we mentioned the consistency. So maybe one way to tackle the consistency is that instead of having universal products that work around the globe, maybe we need to tackle, maybe one region at a time, or maybe we need to be more specific to one commercial variety. On the commercial side, that has a lot of issues, but maybe on the science side, this is what we need to do. We talked about community, and right now what we are doing, we're only commercializing one microbe, right? So maybe another way is to actually think about, can we commercialize a mixture of microbes? We talked about community. Maybe we need to introduce a whole community in order to be able to be successful. So I'm just going to conclude. I think we are, once again, as a microbiologist, I think it's a very exciting time. I think we are on the verge of a new opportunity, and we can really make a technology that has historically been very inconsistent to try to make it more consistent. Thank you. So the question is, what do you think the consequence of the phytobiome, and how can the public sector and the private sector work together, right? So we do work a lot with the private sector. We do work a lot with academia, okay? And I really truly believe that that the industry and the academic need to be involved much earlier, doing kind of like the discovery and the research time. Usually what happens with the industry is we get a call from the tech, from the licensing, from your universities, and they say, hey, we have that technology. Are you excited about it? I truly believe that we need to be way before that. Right, so we need to be involved. We need to help each other understand the challenges and kind of guiding the research in order to be successful down the line. Now, of course, we're going to have to be creative because people need patents and we need to to protect each other interest. But I really truly believe that if we really want it, we can find creative way to do that.
Most of what's done classically in the Serenade, for example, has been isolated and sealed microbes, and that's, of course, the easiest way and has been very productive. You mentioned products where you would have multiples. So how would you approach that? Is that looking for multiple individuals and trying to combine those, or would you be looking at a consortia that you try to identify and culture together? And my second question is, given that there's multiple constituents, do you see regulators with that? As you know, it's one at a time typically, and EPA gets quite worried about multiple AIs and require multiples of time given for each additional microbe.
Okay, so there is two parts to the question. The first part is, right now we are commercializing single microbes. So if we were to commercialize multiple microbes, how would that look like from kind of like a manufacturing point or something like that? And the second point is, what would the regulators say? So from a manufacturing point, commercializing consortia. So there is a lot of talk right now about consortia. So there is a lot of biotech companies that are working on consortia. I think it's very challenging. I think commercializing consortia and manufacturing and making sure, so I talked a lot about consistency, right? We need to make sure that the drug is consistent. So that's going to be a challenge. However, I think at the beginning, when people started working on microbe and commercializing microbe, there has been challenging. So I think that, if you put enough, not power, but enough, people working on that, I think that we will make progress. From the regulatory perspective, I don't really have any, much insight about if that will be a problem. My gut feeling tells me no, but I just might be totally wrong. Especially in the US, microbes are really viewed in a very positive way. I don't see, why the regulators would change if we add five or six to as a product. I know there is some commercial product, out there. For example, Baker Underwood. I don't know if you know that company, but they are definitely, commercializing a soup of, not a soup, but a association of different microbes. I believe they put them one by one. I don't think that they're manufacturing the whole thing. So, it's being done right now. Maybe not to the extent of a consortia. So I just don't see it will be a problem. Mm-hmm. Yes.
Yeah, just now you mentioned, something importantly including the diagnostics to the growers. So I was wondering, what did you need? You need, the industry will provide the service of the universities to do that.
So I think the question is around diagnostics. So I had a bullet point, right, around diagnostic in the field, right? So I don't really have any, much comments if it should be the industry or if it should be the university. I know that, people talk a lot about digital farming, for example. So that's kind of like a big buzzword in the industry, where the industry is trying to do a lot of phenotyping in the field, because we do understand it's a very complex environment. So instead of trying to do that in the laboratory and then bring it back to the field, people go directly into the field. So this aspect of diagnostic, I think we had a talk yesterday around nitrogen, for example, diagnostic. I just don't think it matters if it's the university or the industry. In general, the industry is going to have a tendency to provide that service because they want to sell a whole package, right? So they want to come to the farmer with solutions, and usually they want to have a nice portfolio of solutions. So if they do develop a specific solutions for, let's say, a specific genotype or of plant or a specific soil, they are most probably going to want to associate that with a specific service. But
with relation to that, the package, do you see chemical companies like Bayer getting into the fertilizer industry? Because we know that the interactions between microorganisms and plants includes changes in nutrient cycling.
yes. So Bayer as a sorry, the question, yeah. So the question was, do you see Bayer as kind of as a company to get into the fertilization business? Because fertilization is, of course, a big part of the microbiome or the interactions. So Bayer, as for a chemical, a pure chemical standpoint, I don't believe we'll get into the fertilizer. However, from a biopesticide, from a microbe, we are definitely getting into the area, right? I talked about input reduction, right? Input reduction as well as solubilization of nutrients and so on. So it's definitely a mode of action that we are looking at.
I'm going to have to ask all further questions to come up and ask the speakers. Just thank you very much for your patience in going over in the media. But let's give them a So this concludes our session. If you have any questions for the speakers, come find them. And please, if you're interested in the Phytobiomes initiative, there is a booth. You can visit it and get a T-shirt. Jan will model it here. Thank you. Yeah, I will find out the answer. Yes, yes, it's just exactly what I need. Thank you. Thank you.